Thank you all for coming out on this balmy Wellington evening to listen to my lecture, so I appreciate that. I was reading in the Listener, as I do, a quote from Michael Douglas, who said, "Being a celebrity is like having dementia. Everyone knows who you are, but you have no idea who anyone else is." And so tonight I'm feeling slightly demented because you now know who I am, and I don't actually know who any of you are, apart from one or two who I do recognize. So, but maybe by the end of the evening I'll have a chance to meet some of you, and you can help me feel slightly less demented. I'd like to start telling you about a friend of mine, Steve. This is not Steve, but this is like Steve. So Steve is coming up 60. He's thinking a bit more about the future perhaps than he might have before. He's wondering how long he'll keep working, how long, when he might retire, what he will do with his life when he does retire. He's looking forward to getting the senior citizens discount card on public transport and movies. He also knows that as you age, that people have more the risk of health problems coming up. So he wants to make sure that when he does retire, he's still healthy enough to actually enjoy his retirement. And he's starting to worry a little bit because he's starting to forget things a little bit now and again. He doesn't know whether he should worry about this or not, but he's heard about Alzheimer's disease, so he's thinking, maybe this is coming, maybe not. How would you know? And what would you do? So that's Steve. And Steve is not alone, because in 2011, the Southern Cross did a health and lifestyle survey of New Zealand. And there's various things that were categories of information they were collecting: healthy lifestyles, bad lifestyles, but also people's fears and worries. And, well, the biggest fear and worry was people's weight. 80%. But the next biggest worry out of this whole survey was losing memory with age, 50%. That was more of a worry than financial security after retirement, after becoming ill, leadership of the country, who cares? But losing our memory when you get old is a very significant problem. People are aware that this happens, and no doubt you're here because you're very aware that it's very commonly happens with age. And you and Steve are not alone, because we have an aging population. And this is a little pie charts here showing the percentage of the population in different age bands. And you will see that in this light blue, which is the 65 plus age band, 2006, about 1 in 8 people in the country were aged over 65. In 30 years, over a quarter of the population will be over 65. And I'll be in that group. If I'm still around, I'll be in that group. So there's a very significant changing population demographic going on, not just in New Zealand, but in Western countries generally. So all Western countries are seeing the same trends. Well, there's nothing wrong with aging. There's a lot of healthy things going on during aging, and you're accumulating wisdom and maturity, keep going as you go. So, but also we know that the risk of significant health problems does also increase with aging. So it's of significant concern to individuals and to society because of healthcare costs, stress, and costs to caregivers and families and so forth. So a very important problem coming up. If we look at dementia, can you hear me if I walk around a little bit? I don't like to be stuck behind a lectern. Can we hear okay? Okay. If we look at dementia, based on statistics from Statistics New Zealand, about 1% of the population in 2008 suffered from dementia. The projection is, and as you can see, an upward curve that's getting steeper. So in about 3 decades or so, almost 3% of the population will have dementia. So That's if nothing changes, nothing else happens in the intervening period. Right. So this is why it's called a tidal wave, an epidemic. Various terms have been used. And of course, dementia is not the only issue. Lots of health problems have an age-associated risk, from hearing loss, visual problems, balance problems, physical frailty, cardiovascular problems. But dementia is, I think, a particular, concern to people because it changes them. And it changes their personality. It's quite different from cancer, let's say, or heart disease. And it can also be quite prolonged, right? Years, decades, you can have gradually worsening dementia. So it's scary. So by 2050, it's projected that New Zealand will have about 150,000 cases of people with dementia, and annually, coming forward, 45,000 new cases per annum. So you can see there's a significant problem. And no doubt you've heard about this in general, if not the specifics, that there is this coming issue. And so there's a lot of, not quite panic, but significant concern at all levels from the government down, because there's huge issues here, costs and so forth. So rather than going through all the huge issues with you, I thought that I'd come back to the title of my talk here and sort of try to put a slightly more positive spin on the whole issue and try to give a little bits of possible good news along the way in the face of this coming tsunami of dementia crisis situation. Okay, well, here's a piece of good news. 15,000. Give me a guess what this number is representing. It's actually probably an underestimate. This is the number of words, English words, that you know in your vocabulary. And it's probably, like I say, an underestimate. So your brains are doing quite well. You've got a fantastic vocabulary. And the interesting thing about aging is it's one of the things that gets better with age. So your vocabulary is improving. It's not quite every page on the Oxford Dictionary, but it's actually improving. And no doubt your skills at the crosswords are improving as well. Okay, you can probably beat the pants off your kids doing crosswords because you're better at this sort of thing. So here's a piece of good news. You can take this home with you, put it in the bank. Your vocabulary is getting better, particularly if you keep reading and keep the vocabulary usage going. So that's a positive. Details. So I just want to come back to the issue of learning and memory and just briefly discuss why this is important. Of course, what is learning? Well, learning is changes in the brain that takes place as a result of your experience, whether it's some kind of training, whether it's incidental learning, whether it's your experiences during daily life. But changes in the brain occur, information stored, and then it changes your behavior. Okay? If there's no changes in the brain, there's no learning. Okay? The brain undertakes this task, and it has to change in order to learn, and then by having changed, you can now express altered behavior, and that's how we know people remember things, because the behavior is different second time around than it was first time around. And why is it good? Well, it's important because you need to tell the difference between what's familiar and what's novel. The brain loves novelty. The brain seeks out novelty. The brain pays great attention to things that are novel because things that are novel could be important, and you need to decide whether they're important or not. Okay, so they may be important to remember, they may not. But how do you tell something's novel? Well, you can only tell if you know what's familiar. So you have to know what's familiar to know what's novel. Okay, so the brain has to make these calculations. And that takes memory, obviously. To know what's familiar, you have to have learned. It's important to know what things go together. When you meet someone, you see their face, you hear their name. Well, take me, for example. All right, you've been introduced to me. You've heard a little bit about my background. You see my face. You hear my voice. So you know something about me. And later on, when you see my picture in the, I don't know, in the crime page or something, you'll think of that voice. Gosh, I remember that person. So you learn the relationship between various stimuli, and you put them together, and you mold your representations of the world, people, places, things. They all have multiple characteristics, and they get bound together. Okay? This room becomes part of your memory store for this event. Okay? The people next to you, the banner, the screen. You also have to be able to adapt to the consequences of your actions. If you've had kids, you know about how to train kids up in the right behaviors. And you have to teach kids that there's consequences to your actions. Okay? If you touch a hot plate, you're going to get burnt. And if you couldn't learn the consequence, the association between your action and the outcome, you're going to be in trouble. You won't survive. And so this is another kind of memory that you have to learn. So associating things is a fundamental part of learning information. And it turns out that this is important for all cognitive functions. It's important for your thinking, for your language, for your interactions with people, your social interactions. It's important for all your motor skills. It's important for your creativity, even for your emotions. So it's a fundamental building block of all cognitive activity. Okay, and any animal that moves around has to be able to learn to move and learn and adapt to their environment. So it's fundamental. And if the memory function gets impaired, cognitive ability becomes impaired as well. Having said that, you don't need to remember everything. You don't want to remember everything. You don't need to remember the sounds of the ventilation system, right? Who cares about that? You might pay attention to it briefly, but then you adapt to it because it's not important. It's no longer novel. It's not interesting, not relevant. You don't need to remember it. Right? Many other stimuli around here is the same. So we don't need to remember everything. And also, we do forget things from time to time. Everyone does. I mean, people worry about forgetting things when they get older, but think about teenagers. My God, they're forgetting things all the time. You ask them to do something, they never do it. Right? So memory's not perfect. It's not a videotape of what's going on in your life. You can just play back whenever you like. So it's a biological system, and sometimes it works perfectly, and sometimes there's always little issues. But just because you don't remember something doesn't mean that you have dementia coming on. All right? If you have a memory problem, it doesn't mean you have dementia coming on necessarily. It could be that you're not sleeping well. It could be that you're depressed. It could be that you're taking drugs for some other thing that's causing some cognitive problems. There's all kinds of reasons why memory doesn't go well. So Steve, just because he forgets things now and again, probably is forgetting that he forgot things when he was younger as well. But it's also, it could be for any kind of reason. And it might not have anything to do with coming dementia. Okay. Well, here's a picture of the brain. I like to draw the analogy between the brain and a cauliflower. It does have a kind of cauliflower features. It's got all these bumps, kind of nice big fat bumps that kind of push up against each other. There's hardly any gaps. And it's beautiful. Well, to me it's beautiful. I don't know, maybe to you it looks kind of weird. But this is a top view. Okay, that's the front, that's the back. The two hemispheres, as you can see, that's the cortex. That's the huge, large part of the brain that the humans particularly have a lot of. Here's an individual nerve cell. That's one of the 90 billion nerve cells that you have in your brain. So you have a few. And if you cut the brain in half and look at the sort of inside half, you can see it's actually quite complicated. Underneath the cerebral cortex area, there's a lot of complicated structures in there as well. So it's actually well, it's complicated. It's anatomically and, of course, functionally. It's only 1.5 kilos. And yet it requires 20% of your cardiac output of the blood supply to keep it running and humming. Okay? So obviously extremely important piece of tissue in the body. If we look at someone who's passed away from Alzheimer's disease, I draw the analogy to a walnut. Because now the gaps between these ridges is very wide. There's been a lot of cell loss, neurodegeneration. Can lose up to a third or so of the mass. So you're down, so you might be down to a kilogram. That means you've lost maybe 30 billion neurons out of your 90 billion neurons, if you do the maths. So if someone's getting dementia, it's not surprising if there's that amount of cell loss. Okay, for those of you who don't know much about Alzheimer's disease, of course it's a form of dementia. That means severe cognitive difficulties in all domains. But it begins with typical symptoms, and people are particularly cognizant of the mild memory complaints that get kind of worse and worse. But there may be other symptoms as well. They might have gradually slowing gait. People have some difficulties with smell, but that happens with a number of neurological disorders, so it's not distinctive for Alzheimer's disease. So it's got a very slow, insidious onset, progressive deterioration, and of course there are many neurodegenerative diseases, and so in order to tell that it's Alzheimer's disease, you have to rule out other factors, like maybe the person has had multiple strokes, cardiovascular accidents, and that might be also causing brain damage. Here's the man who discovered that Alzheimer's is a disease, not a psychiatric disorder, because he could find the brain pathology that's associated with these kinds of conditions. Really, you don't know for sure until you've done post-mortem confirmation of the brain tissue and look at the neuropathology of the brain. Although, having said that, I've added in red number 6 because that's changing. We're now getting, having an understanding that there's various biomarkers, biological indicators that people have the disease even while they're alive. So fancy scanning techniques, sampling of proteins in the cerebral spinal fluid, like through a spinal tap and that sort of thing, are methods that we can use to track that people actually have the disease. Although these are quite expensive and not routine methods, but can be used in research. If you want to learn more about Alzheimer's disease, I recommend that you go to these websites. This is the Alzheimer's Association, kind of international organization based in the States. This is the website for the Alzheimer's New Zealand, and which is based here in Wellington, I believe. So these are good sources of information, further information. So what are the risk factors for Alzheimer's disease? Age, single biggest risk factor. There are some familial forms of Alzheimer's disease. In other words, maybe 1% have known gene mutations that lead to early onsets, 40s or 50s. Okay, this is what we mean by early onset Alzheimer's disease. Okay, so there's 3 genes in particular where a variety of mutations can lead to this early onset Alzheimer's disease. But this is a very small percentage of all Alzheimer's disease. Almost all of it is what we call sporadic or late onset Alzheimer's disease. There is another gene that has some relation to this. This is called the ApoE gene. And if you have the E4 type of this gene, it gives a 40 to 50% risk of developing Alzheimer's disease, but it's not as causative as these ones. So it can't be diagnostic. It just makes it more likely, but not necessarily that you would get the disease. Here's a bunch of other risk factors. High blood pressure, high cholesterol, smoking, diabetes, repeated head injury, Down syndrome. My youngest brother has Down syndrome, and so he's aging like me, and just probably beginning to tick over and show some signs. But people with Down syndrome, if they live long enough, are a higher likelihood. The other thing to note here, which I put in red, is a number of these risk factors are modifiable. You don't have to live with high blood pressure. You don't have to live with high cholesterol. You don't have to be a smoker. These are things that you can change that can modify risk factor. Right? You don't have to take up boxing and have repeated head injury. Right? So these are things that are modifiable, and that's, in a way, part of the good news. So what are the big issues in the Alzheimer's field from I suppose, a research perspective? in terms of the biomedical research area. I'm not thinking about dementia care and that sort of thing, which is also another big issue. But the ones that are focusing on my mind are, one, can we prevent or delay the onset of Alzheimer's disease, for example, which is the most common form of dementia? If you're going to get it, can we detect it early? So we can identify the people at risk and the people themselves can know. And then, of course, can we have an effective treatment? So those, to my mind, are the big three things that are a kind of interest from this kind of research perspective. So let's start with the first one. And now I'm going to try to give you some of the good news, apart from the fact that you have fantastic vocabularies and can do great, really good at crosswords. I'm going to try to give you some more good news. So I'm going to do some studies. Most of these are some overseas things that I've come across in my reading, but also, as you'll see, we'll come across some of the work that's being done at Otago as well. So here's a study. 2008, in the USA, a nationally represented set of people, several thousand people, I believe, of people aged 70 plus. 1993, when they did the study, 12.2% had some form of cognitive impairment. Not necessarily dementia, but including dementia, but maybe some kind of mild cognitive impairment as well, perhaps leading to dementia. So cognitive impairment. 10 years later, with a new cohort, but the same age and matched as well as possible for lots of variables, did it again, cross-sectional study, 8.7% had cognitive impairment. So who knows why? They can't say why, but their thoughts on this was that A, better education was protective against cognitive impairment and helping delay, and that generally speaking, the combined impact of recent trends in medicine, medical factors, lifestyle, demographic, social factors have been positive for cognitive health. It's not to say that no one's going to get Alzheimer's disease, but it looks like it's maybe delaying or pushing back when it might occur. A very similar study was done in the UK. This was published just recently in Lancet. So now this study was done across 2 different cohorts of 8,000 people, age 65 plus. Now they were looking at dementia in this particular study. And these groups, they did 2 groups approximately 20 years apart now, between 1889 and 1994, and then 2008 and 2011. Well, the prediction based on the 89 to 94 study was that in 2011, about 8.3% of the population of this age group would have dementia. And what they found was only 6.5%. So about a quarter less. So that sounds good to me. I'd like to be in that little difference between the 8.3 and 6.5. And again, they can't say why, but in higher educational level, probably better prevention of vascular disease, which is a compounding factor for Alzheimer's disease, such that later born populations, so now we're talking about people who were born in the 30s compared to the people born in the 1950s, maybe a decade out or whatever. But 20 years apart, the significant differences in what's happening over the course of their lifetime, so that by the time they're older, they have less prevalence of Alzheimer's disease and other dementias. So pretty interesting. Now, we talked about modifiable risk factors. Here's another study from the USA. I did a long-term survey of people, over 5,000 people involved in this. This is back in the States. They looked at people in 1994 and looked at the level of exercise participation that they were undertaking, measured at baseline, and then they followed them up at 5 and 10 years later. By 2004, they came to this conclusion that if you control for age, sex, education, baseline score on the cognitive test, diabetes, and hypertension, so you're controlling for lots of different variables, that the more different types of exercise that you undertook was associated with the delayed onset of cognitive impairment, and that if you had exercise sessions, even if you're using just one type of exercise, but the more you had lasting more than 20 minutes, the longer to cognitive impairment. And we're not talking about like going to the gym and lifting weights and, and doing the Zumba or whatever. We're talking about like going out and gardening or yard work, or but it could be going for walks or bicycling or something. So so perhaps not a huge delay in terms of time, but detectable. So modifiable. I just came across this the other week. This study was published last year. I just came across it. It's a study out of India, actually, that bilingualism delays onset of dementia. So they looked at 648 attendees to a memory clinic in Hyderabad in India, and the basic result was this. Compared monolinguals with bilinguals, there was a 4.5 years delay in the onset of dementia. Independent of level of education, or even if they were literate, whether they could actually write. Okay? And it didn't matter whether they were immigrants or locals, as long as they had two languages that they used fairly regularly and were conversant in. All right? So really exercising the brain, you might think, as a result. And that was true for all types of dementia lumped together, but if you looked at them individually, if it was true for Alzheimer's, statistically true for frontotemporal and vascular dementias. So start learning another language. It's my advice. Okay, so in terms of prevention and delay, that's some of the good news, that it's not inevitable. I mean, of course, we're going to die of something eventually, right? But you can delay the onset of dementia through addressing some of these modifiable risk factors. Keeping your brain active, keeping your body active, keeping good cardiovascular health, and so forth. And you can start now. You don't have to say, well, I wasn't doing it when I was 18, so there's no point. All right? You can start now, and it can make a difference. Okay, let's talk about early detection. So here's a graph from a study that, as I talked a little bit about biomarkers before, and how now there's ways of detecting the disease as it progresses. And so this is a timeline here. So if we look at, if you're thinking about individuals, I suppose. So you're cognitively normal, and eventually you start having a bit of memory problems, and we call that mild cognitive impairment, or MCI. And eventually, things get worse enough that you get clinically diagnosed as having dementia. Okay? And so here are the memory tests here that are kind of picking up. These are just little representations of the memory tests that are just coming off baseline enough that you can then be diagnosed as MCI or then diagnosed as having dementia. But there's various scanning techniques, for example, looking at the proteins that we know are important in the etiology of Alzheimer's disease, using either PET scans or MRI scans. You may not have heard of PET scans, but MRI you'll probably be familiar with, looking at the structure, the size of, for example, of a brain area called the hippocampus. It's actually an area I work in. And these start showing signs. As you can see, they're taking off well before a person gets classified as having MCI. And this period of time could be 10, 15, 20 years. All right, so the disease has an onset over quite a lengthy period of time. Now, some people think that's quite scary. In other words, you could be sitting there and you could have Alzheimer's disease and not know it, and but it's coming, but you wouldn't know. So in a way, that's kind of scary. But on the other hand, think about the opportunity this represents, this huge window of opportunity for treatment if we had good treatments. In fact, we might have good treatments and not know it, because the treatments that are used, and I'll come to this in a moment, are only used when people already have Alzheimer's disease, right? So maybe it's too late. If we could give the treatments earlier, when we knew people actually had the disease earlier, we could maybe even slow it even further. So I'll come to that a bit later. But anyway, the point here is all these tests, basically all these tests that are used in a research sense for identifying early Alzheimer's disease are very specialized, expensive pieces of equipment, in some cases invasive, and can't be done on a routine kind of testing basis out there in the real world, in the general public. So there's a desperate need to find and identify biomarkers with some more useful way of doing this. And so then I'd like to now introduce my group. I say my group, it's not really mine, but I am the director of this research program within our center, but this is a smaller group within our center who's specifically working on a grant from the Health Research Council, as Rodney mentioned, on cognitive decline during aging and Alzheimer's, biomarkers and therapeutic molecules. And I'm going to show you a little bit of the research on biomarkers. So the idea that we have is that maybe we can develop a blood test. Maybe there's things happening in the body that kind of show up in the blood, and there's changes in molecules in the blood that we might be able to detect and at an early stage of the disease. So I'm going to give you a little bit of data, really pretty much hot off the press, not yet published, but we think it's pretty exciting. So we gathered up blood samples from people who either have been diagnosed as having Alzheimer's disease or age-matched controls. And we have a little over 40 of each. We've got the blood samples, we've stored them in a biobank, and we've started analyzing them. And then the idea is, if we can find some molecules that distinguish between AD and control, we can then try to see if those same biomarkers can be seen at an early stage of the disease and predict outcome. All right? Can they be used as predictors of getting a diagnosis of Alzheimer's disease? So that's the holy grail, really. Identify biomarkers and then see if they are predictive in people who don't yet have obvious signs of the disease. Okay, well, we have a specific class of molecules that we've been studying called microRNA. I don't want to actually go into any technical details about what microRNA are, but they are a form of RNA, if you know anything about genes and the DNA. DNA, of course, codes for proteins, and it goes through an intermediate step by making RNA, which then gets read out to make proteins. In microRNA, are very small kinds of RNA that don't make proteins, but they have other regulatory functions. They play a role in regulating proteins and also gene expression. So we had a whole panel of microRNAs that we were investigating in these blood samples, and we found 3. It took only 3 changes in order to distinguish between people who had Alzheimer's disease and controls. And one of them went up quite substantially in the bloods of people who had Alzheimer's disease, and 2 went down statistically. So these 3 microRNA could distinguish between, with fairly high accuracy, not 100%, but quite high, more than acceptable accuracy between Alzheimer's and controls. So, is it predictive? Well, we're still working on that. But I'm going to show you some preliminary data. So we're going to take this one, which looks extremely promising, and we have some blood samples from people from another study done back in 2002, 2003, and we got the blood samples were taken from them. They were stored, and there was some left over. So we had access to those bloods, and we then we went back and found those people again and found out whether they had developed dementia or not. So it's not a huge number of people involved here, but when we looked at the microRNA, interesting thing came out. So these people, if you have AD, right, you have an increase in the amount of this microRNA. If you're going to get AD, but you don't have it yet, you actually have much less than controls. So this is an indicator of this reduction in this microRNA is an indicator the disease is coming, And then when the disease arrives, it switches over and you have more of it. Not quite what we were predicting. We were kind of thinking, well, maybe it would be also up beforehand as well. But it's quite interesting that there's obviously some dysregulation in this particular molecule, and probably others, which we're yet to identify, still studying. But this one switches from being low to high in the pre-AD stage. So they were cognitively normal at this stage. No neuropsychological evidence of cognitive deficit. They were chosen because they were cognitively normal for this study. And yet, some of them did develop Alzheimer's disease later on. There's a little red arrow. That's great. Now, we also do a lot of work with animal models because there's very little experimental work, of course, you can do with humans. Not many people putting up their hands to kind of have their brains dissected or whatever. So we have to do some work in experimental animal models. I hope you can understand the importance of doing this because we can't do a lot of the things we need to do to understand the disease. So we also have a mouse model of Alzheimer's disease. Now, let me tell you something about mice. It's great to be a mouse because mice don't get Alzheimer's disease. Their genes are slightly different, and they can get as old as they like. They don't get Alzheimer's disease. So if you're going to have and rats would be the same. So if you're going to have a mouse model, we have to give them some of the genes that we know in humans will lead to Alzheimer's disease. So this has been done. And there's some strains of mice now that have the human genes, one or two of the human genes, that will lead to Alzheimer's disease. And sure enough, these mice start developing with age. They develop the pathology, behavioral deficits, and so forth, as if they had Alzheimer's disease. It's not exactly the same. It's not exactly Alzheimer's disease, but it's a good model. So we have some of these mice in the lab. And we took that same microRNA, and we looked at it when they were 15 months old, which is quite old for a mouse. Not senile yet at this stage, but quite old. And you can see that the levels of this microRNA that was up in the humans was also up in the mice that have this disorder. And look at this. The same thing's happening to that microRNA before they have the problems. They have much less of this microRNA. So this is a way of validating both in the animals that are going to get the disorder and the humans are showing the same pattern of effect. We think this is quite exciting. And so we're madly trying to kind of pursue these studies and nail it down, find out what other microRNA are important and so forth. So, so this is some good news. I think this is the good news of this is that people are working on the problem, and we think we're making some progress. Okay. So let's then go to treatment. I want to tell you a little bit about what's currently out there, and then I want to tell you a little bit about what we're trying to do to amplify. Okay. So FDA, who knows what the FDA is? Food and Drug Administration in the United States, the kind of the master puppeteer of basically all drug approvals in the Western Hemisphere. Europe tries to go it alone, but in the end, it always keeps coming back to the FDA. So there's three drugs that are approved by the FDA that have very similar mechanisms of action for Alzheimer's disease. There's one more I'm going to tell you about in a moment. But these three have similar mechanisms of action. They all work by blocking the breakdown of this particular neurotransmitter called acetylcholine. And so because acetylcholine levels go down with aging and dementia, and so the idea is to prop it back up with these drugs. And what does acetylcholine do? Well, it does many things, but you can think of it a bit like a natural caffeine for the brain. It's one of the things that sort of keeps the brain kind of tuned up, you might say. And you'll be more alert and attentive and so forth when you have more acetylcholine floating around. It's not necessarily a specific memory molecule as such, but it can help the memory system work better because cognition is better generally. And about half of the people who take these drugs experience a modest improvement in cognition. They don't stop the course of the disease, however. Oh, I want to say one thing about this. One of these, donepezil or Aricept, is the only one of these that's approved by Pharmac for subsidized delivery in New Zealand. The other one is called memantine. A trade name is Abixa Namenda, approved for the treatment of moderate to severe AD. So this is the same for all these drugs. They're approved for the treatment treatment of moderate AD, after people already have Alzheimer's disease, because that's how they do the clinical trials. You find people who have Alzheimer's disease, and you give them the drugs and see how it works. Because you can't identify who the people are before they have the disease, it's hard to do the drug trials, at an early stage, because identifying the early stage is very difficult to do. Mementine, actually, the actual mechanism of action of this is not entirely clear because it's quite a complex molecule. But one of the things it does is probably protect cells against toxicity arising happens when nerve cells become too highly activated. So actually, too much of activity is a bad thing, and cells become damaged if they're overactive. You can think about epilepsy as an example. You can have brain damage from having too much activity. Okay, well, there's some more good news. There's some recent studies. So basically, you're given Aricept. It may or may not work. If you have side effects, because they all have side effects. If you don't respond well to one, you might try the other. So then you go around and try out the drugs. This study, again a fairly recent study, decided, well, let's give people 2 drugs. Let's give them one of the acetylcholine breakdown inhibitors, and let's give them mementine, and look at cognition. So this is a 4-year study, which suddenly disappeared. We'll try again. This is a 4-year study. Sorry. And so here's a kind of dementia score, and this is a number of errors. As you can see, that errors are going up as time goes on. So the people are getting worse in their abilities on this scale. If they received one of these drugs, in this case Aricept or donepezil, you can see it produces a small improvement in cognition at year 1, but then the course of the disease is pretty much parallel. So it hasn't really stopped the course of the disease, it's just raised cognition a bit, so then it just stays a bit better right through. Then they gave both drugs, mementine and donepezil, and you can see that not only has it made it a bit better, but it slowed the course of the disease, of the symptoms. So maybe some kind of polytherapy could be a useful strategy going forward. They also looked at not just dementia, they looked at independence, the ability to live independently. And you can see that the level of dependence increases quite markedly over the 4-year period, from 20% to 80% over this 4-year period. And it's not really actually affected by the single drug by itself, by the acetylcholine inhibitor. But if you put the 2 together, it did slow the rate of becoming more dependent. So, the disease is still happening. We're not curing the disease. We're not stopping it. But at least it's providing benefit. And of course, the quality of life issues are big here. And if you can just, as we talked about, delay the course of the disease, that's a significant benefit. This needs to be replicated. I mean, this is just one study. People have to do this many times to kind of confirm, but it's something to think about. So unfortunately, no drug stops the disease. We need more science to understand the causes, to devise rational treatments aimed at disease mechanisms, and for the early detection of the disease, to identify biomarkers. So if I still have time, I have time. I'm going to tell you a little bit about now what we're doing. And just coming back to this point, to devise rational treatments aimed at the disease mechanisms, you need to know what the disease mechanisms are. Okay, so there's a lot of understanding now of what some of these disease mechanisms are underlying Alzheimer's disease. And I'll tell you one of the main hypotheses. It's actually about proteins gone bad. This is actually true of just about every neurodegenerative disease you can think of. Parkinson's disease, Huntington's disease, frontotemporal dementia, supranuclear palsy, the list goes on. They are all protein gone bad diseases. You've heard about mad cow disease. So that's a protein gone bad. Starts aggregating, clumping, becoming toxic. Each disease has its own proteins that are doing this, but they're all very similar. And Alzheimer's disease is no exception. So here are, in the neuropathology postmortem tissue, if you look into the microscope, you can see 2 kinds of main changes neuropathology. One is these extracellular, what are called plaques, these black things called senile plaques. And they arise from are kind of triggered by the accumulation of this small peptide, which is just a small protein, called amyloid beta, which is a core molecule. Then there are lots of other molecules in here as well, but it seems to be triggered by accumulating this amyloid beta peptide. The other protein gone bad is tau, the tau protein inside of cells, and it starts to aggregate also. There's changes in the kind of structure of the protein. It starts to clump, can't be broken down, and it builds up and causes what are called tangles. And you can see these nerve cells. If you remember the nerve cell that I showed you before, near the beginning, this looks nothing like it. It looks sick, and it is because it's filled with these tangles of the tau protein. So many people now believe that the disease etiology arises primarily from this amyloid accumulation. Amyloid comes from a protein called, it's not very creative, but it's called amyloid precursor protein. So this is a long protein which stretches, this is the cell membrane, and this is the inside of the cell, and this is the outside of the cell. This is one of these proteins that goes across the cell membrane, stretches across. Part of it's on the inside, a lot of it is on the outside, part of it's gone through the membrane. And this precursor protein, it's called precursor because it can be snipped up by hungry enzymes, in this case called beta and gamma secretase, which can clip the protein around these positions and form this smaller, much smaller molecule. Watch this. Check that out. And that's the amyloid beta, that represents the amyloid beta protein, or peptide. So everyone has it. Everyone has some normal amount. But during the disease, it starts to accumulate abnormally. And when it does that, then it starts to bind to each other. I told you how it aggregates. So then you get 2 of them, then 2 make 4, and then 4 make 12. And so it builds up, and eventually this soluble protein, which probably has some helpful normal function, actually builds up and eventually forms these plaques. And it's thought that the plaques then lead to the stimulation of this abnormal tau accumulation and also inflammation in the brain, which is also a bad thing for neural function. And so this is kind of the triggering event. So this has stimulated a whole lot of research aimed at trying to stop this process. So the research has several strategies. Obviously, none of which are 100% successful yet, but this is what people are thinking about and working on. And of course, drug companies are quite interested in this sort of thing as well. So first of all, you might say, well, this will be easy. We'll develop drugs that block either the beta or the gamma secretase enzymes. And Bob's your uncle? That's an American expression. Sorry. And then you will prevent the formation of amyloid beta. It turns out that unfortunately, beta and gamma secretase don't just work on the amyloid precursor protein, they work on other proteins as well. So you get side effects from impairing the normal function of these enzymes on other proteins as well. So now drug companies are working on what are called 2nd and 3rd generation drugs that could be specific for the action of these enzymes on this particular molecule. So that's an area of current research. Here's another one. Why don't we just make a vaccine? Got vaccines for all kinds of diseases. In principle, you could have a vaccine for Alzheimer's disease. You inject antibodies or get the person to develop their own antibodies to this amyloid peptide, and it could just soak it right up like a sponge, and this might be a way of preventing the development of Alzheimer's disease. So there have been clinical trials on this. Unfortunately, initial trials had, again, side effects, and unfortunately, a couple people got some meningitis whether it's because of the treatment or not, you don't kind of know, but they got form of meningitis, and so the trial was stopped. But there was some cognitive improvement while the study was going on. So again, now they're trying to work on second and third generation vaccines to try to kind of get around these problems. But gee, wouldn't it be nice? You get an injection, get a vaccine, away you go. Maybe you have to top it up every 5 years or something. But so that's exciting, but still undeveloped. There's also people, particularly in Australia actually, who are quite interested in trying to find drugs that prevent the aggregation. So trying to stop that process from happening. And then you could just say, well, we can't stop the actual process, but if we can just counteract the toxicity of the aggregated protein, maybe that would be a good thing. And actually, that's the kind of direction that my research has been going in this program of work. So let's come back to the amyloid precursor protein. Well, it happens that there's another hungry enzyme out there that can also clip the SAPA molecule, and this is called alphasecretase. And alphasecretase clips, as you can see, in the middle of the amyloid beta sequence. So if alphasecretase gets to this protein first, it prevents the amyloid beta from being made from that particular protein. All right, so now we end up with this larger fragment, which is called secreted amyloid precursor protein alpha. So we'll call this SAPA for short. Okay, and then this little fragment here is inactive. It's too short to be disease-causing. So you might say, well, maybe we should try to get more of this. And you think, well, why should we get more of that? Well, actually, first I'll tell you that in Alzheimer's disease, as the levels of amyloid beta go up, the levels of this protein go down. Okay, because they're competing for the same protein. Either you get one or the other, but the same protein can't give you both. So there's a sort of a competitive element to this. And why do we care? Well, it turns out that amyloid, this SAPA molecule, this longer fragment that gets secreted, is neuroprotective. It promotes growth and formation of new synapses. It facilitates memory changes at the synapses. It enhances memory. It's naturally occurring. So we think that this could be a useful target. And so we think that increasing the amounts of some of these protective molecules, and this is only one that our group is working on, but the one I'm just kind of mentioning, like the SAPA molecule, in the brain will help treat aging and disease-related cognitive decline. That's a nice hypothesis. Who would like to volunteer to be treated? Well, we have some mice who have volunteered. So, again, this is the value of having a mouse model now. We can try out our ideas before you go to a clinical trial. In fact, you have to try out these ideas in these animal models before you can go to a clinical trial, if you're not allowed to. We have to undertake this basic research first. So here's an experiment which is basically built on our understanding that learning and memory requires changes at synapses. When you learn, the synapses that are involved in representing that information, that are connected with each other, they're active at the same time, they're sort of coactive, and they strengthen their connections. Okay? And the whole point of this is that later on, when you come to remember my talk, you don't have to kind of remember everything. You just have to remember my beautiful face. And the whole talk will come flooding back to you. All right? You only need partial information in order to retrieve. And that partial information can retrieve the whole experience because things are so well connected with each other now. So this is the idea. Okay, so we can study, this is called synaptic plasticity. And we can study synaptic plasticity in the lab by using electrophysiology techniques, which I'm not going to go into the details, but we can monitor the strength of the connections, synaptic connections between two groups of neurons with these techniques. So here's a little experiment that's run over a 90-minute period, and here's so we're measuring the electrical waveforms we get when we do these experiments that are the electrical signature of the synaptic transmission. And there's a baseline period, and you can see it's basically not changing for 30 minutes. Then we give these synapses an experience, and the experience is just a bit of kind of much higher frequency stimulation, but very short. And why do we do higher frequency stimulation? Well, that's what happens when you learn. Cells become more active, all right? The ones that are representing that information. So we do that electrically in these synapses, and you can see now that the strength of the responding jumps up. And it stays up for these 90 minutes. And in other experiments, we've shown that these changes can last for days or weeks or months. So there's a lot of evidence. We could have a week's course on this, but there's a lot of evidence that these changes are a critical part of learning. So it's a kind of a model now of the memory system. Well, so in these mice, we did this experiment in these mice, and in control mice, little mates who didn't have these genes. The ones who did have the genes, at about a year of age, they start having these plaques. So now the plaques are in red. So these are just like the plaques that you see in the human disease. And the green that you can see, if you can see it, indicates the inflammatory cells that are kind of hustling to the rescue to try to get rid of these deposits that are not normal. Unfortunately, these rescue cells are themselves causing more impaired neural function. So it's supposed to be helpful, but it's also hurtful. Okay, so the mice that have in the green, which you can hardly see, is the control amount of synaptic plasticity or potentiation of the responses. The mice that have the genes, that have the disease, the disorder, I should say, have much less of this ability to strengthen the synapses. They also have impaired behavior. Now, we did a bit of gene therapy with these animals. We injected them with a virus, an inactive virus, but that contains the gene for SAPA. Okay? So I want to emphasize, this is not a real virus. This is just using the virus packaging system. It's not an active virus. But it's inserting the gene specifically for this protective molecule. Whoop, come back. For this protective molecule, SAPA. And it also has a fluorescent protein so that we can tell that it's actually worked. So this is an area of the brain, it's called the hippocampus, I mentioned before, where these cells are now expressing this fluorescent protein and also this SAPA. All the controls also have the fluorescent protein, but only this group has the SAPA. And look, the group that has the SAPA, the brown triangles, they have exactly the same amount of plasticity capability as the control mice. So I'm not going to stand up here and tell you we've cured Alzheimer's disease, but I'm going to tell you that we have some pretty exciting results that is inspiring us to carry this forward, because we think this is very promising. We've already now replicated this effect in another study. So that's, I hope, some more good news based on research in terms of what we're doing down there at the University of Otago. Or should I say up there? Depends on your worldview, I suppose. So there's prevention, early detection, effective treatment. These are all the big issues that I think we can say there's some good news on each level based on the research and the understanding of the importance of lifestyle and so forth. Unfortunately, it's not enough. There's not enough good news. We can't say that we've been able to stop the disease or prevent it from occurring. But I think we're on the track. And of course, it's not just us. It's a huge international effort. Be reassured, it's a major field of research in neuroscience. It's just difficult, but we're slowly making progress. So I want to finish by thanking the sources of funding for our research. The first two are government agencies. So your tax dollars, my tax dollars, going to the government, come back out through the Health Research Council and the Royal Society of New Zealand Marsden Fund that have helped provide a lot of the research funding for what we do. We also want to thank a private trust, which is the Neurological Foundation of New Zealand, who are actually having their fundraising campaign this week, actually. You might be seeing some of the information there. So they've been an important source of funds for us as well. But I must say, getting these competitive grants is quite difficult. There's never enough to go around. We're always looking for money, and so we're very grateful that actually there are also public donors out there and people who are able to kind of make either small, large bequests, endowments, whatever, have been coming to our center and for, in part for this kind of research that we're doing. And so we're very grateful for those people who have done that. And of course, to the research participants who have been involved in our study very kindly. So coming back to where I started, I'm here not just in a way talking about our work and about Alzheimer's disease, but as a representative of our Brain Health Research Center at the University of Otago. We've been going for about 8 years. It's been a privilege and an honor to be the director for that time. And you know, it's a fantastic thing. There's 40 plus research groups and clinicians involved, including the neurosurgeons, the neurologists, and the people spread across campus from the science departments to the medical school departments. And there's our website there if you want to know more. There's also, we have brochures from about our center that's there on the table there. And you're welcome to go onto our mailing list as well if you're interested, and you can come talk to me about that. And also, this brochure will tell you how, if you want to make some private contribution to our research, how you can go about doing that, which of course be very welcome. The final piece of good news, though, is that now we don't just have our center working on this and individual centers around the country working, but we now have a new Center of Research Excellence that's just been announced by the government as one of the 6 top most important things happening in New Zealand in terms of research. And this is a collaborative effort between the University of Otago and University of Auckland. I'm a co-director with Richard Fall in Auckland. And it's jointly hosted by Otago and Auckland. And it's called Brain Research New Zealand, but it's really more specific than that. It's not all brain research, but it's devoted to understanding the aging brain and aging-related disorders and what we can do about it. So the idea is lifelong brain health. And we're pleased to have some also partners at the University of Canterbury and AUT. So this is just starting. In fact, we don't actually get the money till next year, but we're already preparing. And we're really enthusiastic about this new ability to combine our resources, our strengths, our expertise, our ideas, and work together as a national unit to address these very important issues. So stay tuned for further progress on this front. And so once again, thank you for coming along. I hope I've given you some interesting information. If you're a Game of Thrones fan, winter is coming, but I'd like to think that in this field of research, spring is coming. We're making some progress. Thank you very much for your attention.
Questions? Shall I? Would you like to? Question to start?
Stunned silence.
Debated or? Is there one down the back? Sorry, I said I had one right.
Thanks. You're talking about all the things that we could do to stave off this. I was thinking that more brain connections, more neuron connections seem to be involved here. How does that process relate at all to reduction and extrusion of plaque material? Is there any knowledge about how this actually could work, considering that the brains aren't donated until the subject's dead?
I guess this is where a lot of the animal model research has helped us. I mean, the plaques are first identified in the human brains, of course, but then what's the effect of having the plaques and how they got there basically requires the work in the animals. And basically what it's found is that in some ways, the plaques, strangely enough, might be somewhat protective. Because the plaques are not soluble. They're not biologically active, as it were. They're taking up space. They're causing inflammation. But in terms of toxicity, and so that causes problems. There's no doubt about that. But what we might call the pre-plaque form of this amyloid protein, while it's aggregating into 2s and 3s and 12s and things like that, it's actually more toxic than the actual plaques themselves. So once it aggregates into plaques, it's now kind of inert, except it stimulates this inflammatory reaction. So we need to reduce the toxicity of the soluble amyloid, actually, is one of the key things. And all these, basically all these approaches and strategies that I talked to you about would also work on this process. And the toxicity that's generated does impair synaptic function. It makes the plasticity worse. Synapses get lost. That's actually probably the cognitive decline begins not because cells are being lost, but because synapses are becoming impaired and being lost. And the cell death happens later as the dementia kind of, deepens. But the early loss deficits are probably not due to cell loss, but to impaired synaptic function and synapses being impaired. So this is why education, exercise, and so forth helps build what we might call cognitive reserve, because the brain is better connected. And a better connected brain is better able to survive some loss before it starts to go downhill. There's a very interesting study called the Nun study. You heard of the Nun study? So it's in the United States. Some researchers had access to what is the word for where nuns? Sorry? Convent, thank you. I don't know if they call them convents in the States, but if you know what I'm talking about. So they kindly agreed to perform the study. And the study was, this was basically now older, almost basically retired nuns, and they were going to donate their brains to science. But at the same time, they were being tested and so forth. And the researchers had access to their entry essays when they first entered the convent. Okay? Well, it turns out that a very interesting thing was found. When they did the post-mortem analysis, some of them had dementia, and they had the signs of dementia in the brain. Some of them did not have dementia, still had some of the signs in the brain. And if they went back and looked at the essays, the ones who wrote very elaborative essays and showed higher level of education and ability and had lots of use of adjectives and adverbs and very complicated essays, were the ones who seemed to be protected for longer against the scourge of the disease, and they could survive against the disease longer. They would probably get it eventually if they lived long enough. But this was an interesting study because it showed that it helped develop this idea of cognitive reserve, that there's some people who are better able to resist the onslaught of the disease. So hopefully that's a long answer to a short question, but hopefully that gives you a feel for what's going on. Can we get the microphone over to the... But I think for the recording that they'll need your...
Just from your slides, and you were showing the positive benefits of having diazepam and lamantine, I would wonder how far are we off for individuals being able to have that dual medication and the positive benefits?
So this is a clinical question, and so now I get to now say that I'm not a clinician. I'm a researcher, so I'm going to be hesitant about answering that in any specific way. This was one of the first studies of this kind that I've been aware of. You'd have to repeat this a number of times. That's a 4-year study. So I think before it became widely used, it would be some time. On the other hand, doctors being doctors don't mind doing a bit of experimenting as they go. So it may be possible. Only one of these drugs, remember, is pharmac subsidized. So that's an issue. And I think I think it's probably coming. Of course, the more drugs you take, the more side effects you get, and you've got to balance how this works. It's not that once you start mixing drugs, things, and already people are taking drugs for lots of other reasons. People are taking heart medication and blood pressure medication, and these drugs as well. And people who are getting dementia are taking, they might be getting antidepressants or antipsychotics, and that's making cognition worse. So there's a whole lot of drug interaction things going on. So all these things have to be weighed up and considered before you start kind of piling more drugs on top of each other. This is promising. I will say this. This is promising, but I wouldn't say it'll become routine practice anytime soon yet. But I'd like to think this will inspire more work to just show how well it can be done. Hopefully by my time I need it will be well on the way.
You started off by showing us two large-scale longitudinal studies where in fact the prevalence of dementia in cohorts had reduced over a period of time. I was wondering what conclusions or what implications were drawn from that, and whether there's an expectation that in fact prevalence rates will continue to go down if we continue to live better lifestyles that everybody is urging us to do at this stage.
Thank you, Verna. Good to see you. One of my few faces I recognize. I'm feeling less demented now. Well, first of all, there might be a slight misapprehension. These were not longitudinal studies. These were cross-sectional studies. Okay, so the population at this 70 plus in 93, another population of 70 plus in 2002. So it suggests that there is, for that age group, there is a reduction in prevalence. So how far can it go? Well, we don't know. We know there are all these modifiable risk factors. We know that cardiovascular disease is a really big one. As I told you before, you should take the hint. 20% of the cardiac output goes to the brain. The brain just needs all the oxygen and the glucose to keep going. Right? You can't survive for longer than 5 minutes or something without oxygen and glucose. Unless you're like in a freezing cold river, and then you can survive for an hour or something. But so it's a lot of the. I mean, there's a lot of thinking now that disease is severely compromised and complicated by microvascular problems as well that arise from cardiovascular problems. So these things are slowly becoming better treated and understood and identified. So I think probably it can come down still further. To a certain extent, what we'll be doing is we're pushing out how long people live. The population is living longer and longer and longer. We may be pushing out the age of dementia longer and longer, but then the 70s become the 80s, as it were, in terms of the issue. This is a way of me saying I can't give you an exact number to your question, but what I think there's every reason to believe that you can push out the disease, which is the important thing, with these modifiable risk factors and a better lifestyle, better nutrition, a better attention to your cardiovascular health, exercise, and so forth. And in the end, it means you have a longer period of high-quality life for you and your family, which is, of course, critical. Is that kind of what you're asking? Yeah, thanks.